postponed from 3rd March to 2nd March because I'm not available on 3rd March. So this is live problem solving session for week 6 on NPTEL Bioinformatics Algorithms and Applications course. So in this we'll be seeing about protein tertiary structure because in previous sessions we have seen primary structure, that is protein sequence, followed by that protein secondary structure, and we also studied how protein secondary structure is predicted using the methods such as Chow-Fasman GR information method. And now today we will be seeing about mostly protein tertiary structure. So the contents, so initially I will be discuss about summary. So summary will include protein tertiary structure, about protein tertiary structure and methods to predict the tertiary structure, and as well as classes based on protein tertiary structure, and we will be doing some sample problems. Followed by that, I will take some questions from portal and questions from students. First, to start with, what is protein secondary structure? Protein structure is generally divided into 4 levels, that is primary, secondary, tertiary and quaternary. The tertiary and quaternary structure refers to the 3D and higher order structures of protein respectively. So in this level of organization, tertiary structure of a protein refers to the overall 3D shape of a single polypeptide chain. This structure is determined by the interactions between the side chain of the amino acids that make up the protein. The side chains can interact to a variety of chemical bonds and forces, including hydrogen bonds, hydrophobic interactions, electrostatic interactions and disulfide bonds. These interactions cause the protein to fold into a specific shape, which is stabilized by the interactions between the amino acid. So this tertiary structure of a protein is critical for its function. The shape of a protein determines how it interacts with the other molecules. So the interaction with the other molecules have such as protein to have protein-protein interactions, protein carbohydrate interactions, protein nucleic acid interactions, or protein small molecule or ligand interactions. So any changes in the tertiary structure of a protein can affect its function and lead to a disease. Finally, we have quaternary class. Quaternary structure of a protein refers to the arrangement of multiple polypeptide subunits, that is multiple tertiary chains that make up a functional protein. So in this quaternary structure, the subunits, that is the multiple polypeptide chains, sometimes can be identical or different and are held together by various types of interactions, including hydrogen bonds, disulfide bonds and hydrophobic interactions. So this quaternary structure is mainly important for the function of many proteins, particularly enzymes and signaling molecules. For example, hemoglobin, which is responsible for carrying oxygen in the blood, consists of 4 subunits that are arranged in a specific way to allow oxygen to bind and release. So in summary, the tertiary structure refers to the 3D shape of a single polypeptide chain, while the quaternary structure refers to the arrangement of multiple polypeptide chains that make up the functional protein. So both structures are important for function of a protein and can be affected by any changes in amino acid sequence or environmental factors such as temperature and pH. So now that we have some brief explanation, I have a question. So I'll wait for a minute. The question is, which level of protein structure involves the linear sequence of amino acids? So whether the primary,
Primary structure.
so whether the primary structure refers to the linear sequence of amino acids, secondary structure, tertiary structure or quaternary structure. So before answering the question, again I want to tell on record. So this is a preponement of the class, that is my Monday session on 3rd March. So the same class has been preponed to Sunday. So to provide the answers, people can use the chat box. So I I would like wait for some time because the people who watch the recording they might consider. Okay, so moving forward, primary structure is what linear sequence of amino acids. Primary structure of a protein refers to the linear sequence of amino acids joined by peptide bonds. Even in previous classes, we have. Thank you for the answer. So even in previous classes, we have considered how to calculate molecular weight of a given peptide sequence. So if we consider a molecular weight of individual amino sum of molecular weights of individual amino acids, then we have to subtract number of peptide bonds formed into 18 because peptide bond is a condensation reaction where water molecule is released. So if two amino acids form a peptide bond, a water molecule is released. So how to calculate this molecular weight and how a peptide bond is formed is being discussed in week 4 lectures. So I have one more question. So the alpha and helix and beta sheets are example of which level of protein structure? So whether they are part of primary structure, whether
So here is a similar structure.
yeah, yeah, as already mentioned. Yeah, thank you for your answers. So secondary structure of a protein refers to the local folding of the polypeptide chain into regular structures like alpha helices, beta sheets stabilized by hydrogen bonds. So I have a question based on what I discussed in slide 1. So hemoglobin composed of 4 subunits is an example of which level of protein structure? So whether it is primary structure, secondary structure, tertiary structure, quaternary. That is hemoglobin composed of 4 subunits is an example of. So it refers to the quaternary structure, which is the arrangement of multiple polypeptide subunits in a protein complex. As hemoglobin consists of 4 subunits, each with its own tertiary structure, and their arrangement constitutes the quaternary structure. So there are several experimental techniques used to determine the structure of a protein at different level of organization. So here I have listed some. So the one of the most widely used is X-ray crystallography, but all of these methods help scientists in understanding proteins better, which is crucial for designing new drugs, understanding diseases, and many other areas of biology and medicine. So first, to start with X-ray crystallography method. So it is like taking a detailed photograph of a protein structure. As we all know, proteins are made up of atoms, and when X-rays pass through the crystal made of a protein, they diffract. That is, just they bounce off or change their uh, travel path in patterns. So this bounce off or diffraction is based on the arrangement of atoms in a crystal. So based on this arrangement of the crystals, the diffraction pattern varies, and we record diffraction patterns as electron density maps, and finally figure out or construct the three-dimensional structure. So this X-ray crystallography technique provides high resolution images of protein structures at atomic level. It is often used when researchers need precise details on how atoms are positioned within the protein. But the problem with X-ray crystallography method is crystal construction. We have some methods where crystal construction, that is sitting top and hanging top methods, but the possibility to achieve a good crystal will determine the resolution of a protein. Lower the resolution, better is the protein structure, that is better is the quality. Then we have NMR spectroscopy, so which is like listening to the subtle variations of atoms within a protein. When a protein is placed in a strong magnetic field, radio waves are applied. So the atomic nuclei within the protein resonate at different frequencies based on the environment, revealing the details about the protein's structure. This technique NMR provides the information on interactions between the atoms in a protein and its surroundings. It is particularly used for studying proteins in solution, offering insights into the dynamic properties and interactions. Further, we have electron microscopy, so which allows one to visualize the shape and organization of proteins by bombarding with a beam of electrons. The electrons interact with the protein, generating an image that reveals its structural features. So this technique can provide both 2D and 3D images of a protein structure, offering insights into their morphology and arrangement. So it is especially valuable for the large protein complexes and membrane protein complexes. So then we have the final method. We have one method, that is CD spectroscopy. CD spectroscopy is mostly to study to assess the secondary structure of a proteins, focusing on their helical and sheet arrangements. It works by measuring how differently polarized light is absorbed by the protein, providing information on its folding pattern. CD spectroscopy is sensitive to changes in a protein structure and can detect alterations caused by factors such as pH, temperature, and ligand binding. It is widely used for studying protein folding and stability. Then we have fluorescence spectroscopy also. So fluorescence spectroscopy involves tagging proteins with the fluorescent molecules that emit light when excited by a frequency, by excited at a specific wavelength. By measuring the emitted light, researchers can gather information about the protein's tertiary structure and dynamics. So this allows one to study protein interactions, conformational changes, and environmental effects in real time. So these techniques are essential tools in field of structural biology, enabling researchers to elucidate the complex three-dimensional structure of a proteins and understand their function in health and disease studies. Each method offers unique advantages and insights into a protein structure and behavior, contributing to the advances in biotechnology, medicine, and drug development. Along with advantages, each method has its own disadvantages as well. So here I have a question. So which method involves bombarding protein crystals with X-rays to analyze their diffraction patterns? So thank you for the answers. So the answer is X-ray crystallography, which involves bombarding protein crystals with X-rays and analyzing the diffraction patterns generated. These diffraction patterns provide information about the arrangement of atoms within the crystal, allowing scientists to determine the protein structure. So then I have one more. Which technique utilizes the behavior of atomic nuclei and magnetic field to determine the protein structure? So whether it is electron microscopy, which uses the behavior of atomic nuclei and magnetic field, or fluorescence spectroscopy, NMR spectroscopy, or circular dichroism spectroscopy. So it is NMR, so which utilizes the behavior of atomic nuclei in magnetic field to determine protein structure. So it provides information about the distances between atoms and proteins, allowing determination of its three-dimensional structure. Now I have one more. So which technique focuses us on differential absorption of left and right the circularly polarized light to determine the protein's secondary structure? series. Again, CD, as I mentioned, CD is mostly used to study the secondary changes in secondary structure of the proteins. So this CD spectroscopy focuses on the differential absorption of left and right circularly polarized light by the proteins. So in this section I have so in this I want to talk about X-ray crystallography for some more time, because if we observe any database or number of structures, protein structures known, so most number of the protein structures are through X-ray crystallography method. So because it is a powerful method used to determine the atomic level structure of proteins. So here I have mentioned some general steps involved in using X-ray crystallography to determine the structure of a protein. So first step is purification, crystallization, followed by crystallization, data collection, data processing, model refinement and validation. So the first step is to obtain a pure sample of the protein. The proteins are typically expressed in host organisms, so widely used sorry, the bacterial hosts are E. coli or yeast, and then purified using a combination of techniques such as chromatography, filtration and dialysis. Then the protein must be crystallized to form a regular and ordered array of molecules that can be analyzed using X-rays. Crystallization is a difficult and time consuming process that often requires screening many conditions such as pH, temperature and salt concentration to find the optimal conditions for crystal growth. Once crystals have been obtained, they are mounted on a goniometer and placed in a beam of X-rays. So the X-rays are diffracted by the atoms in the crystal, producing a diffraction pattern on a detector. So the goniometer is rotated and diffracted data is collected at many angles to generate a complete data set. So once this complete data set is corrected, it must be pre-processed to extract the structure of a protein. So this involves several steps, which are scaling, merging data from multiple crystals, calculating the phases of diffraction pattern and building an initial model of the protein structure. So the initial model is refined using various computational tools to optimize the fit of the model to the diffraction data. So this involves adjusting the position of atoms in the model to minimize the difference between the calculated and observed diffraction patterns. Finally, the refined model is validated to ensure that it is of high quality and accurately represented in the structure of a protein. So this involves checking for errors, inconsistencies in the model. So the inconsistencies are clashes between the atoms, unrealistic bond lengths or angles. So now that we know, there are techniques available to generate the structure of a protein, but how to access it? Is there any database of proteins? So here we have protein data bank, which is a database that contains information about the three-dimensional structure of proteins, nucleic acids and other biomolecules. So this PDB is a key resource for researchers in the fields of structural biology, biochemistry, biophysics, and it provides access to the experimentally determined structures of proteins and other biomolecules. So this PDB is maintained by the Worldwide Protein Data Bank organization, which is a collaboration between several institutions, including the research RCSP, that is Research Collaboratory for Structural Bioinformatics, Protein Data Bank Europe and Protein Data Bank Japan, that is PDBE and PDBJ. So the file format used by PDB is a plain text format that contains information about the most of the atom, so that contains information followed by the atomic coordinates of the atom and other biomolecules. So each PDB file represents a single structure and contains information about the type and sequence of the biomolecule as well as the positions and properties. So PDB format provides the Cartesian system based coordinates. So the format of the PDB is divided into several sections, including the header section, which contains a brief description of the structure, and then followed by the atom section, which contains the information about the atomic coordinates and properties of each atom. Then the connect section, which contains information about the connectivity between the atoms in the structure. So to start with the atom section of the PDB, which is main and typically contains information about each atom in the structure. So this has atomic serial number, atomic name, residue name, chain identifier, residue sequence number, x, y, z coordinates of the atom, occupancy. So occupancy is the fraction of time that atom is occupied by the particular conformation. Then we have temperature factor, which is a measure of the atomic vibration or disorder. Then we have then we have connect section, which contains information about the bonds between the atoms in the structure as well as other types of connectivity such as hydrogen bonds and disulfide bonds. So this PDB file format is widely used So now that let's have how this database is maintained. So just go to your Google tab and type PDB. So you see RCSB PDB homepage. Just click. So this is how PDB homepage look. So here they have given option to deposit, search and visualize also. So let me type the widely used one is insulin. So before moving on to that, so now that with the invent of more computational techniques, the data sets in PDB has been divided as structures from PDB, that is structures from experiment, and computer structured models, that is predicted structures. So we have 2 lakhs 32,059 structures from experimentally determined. So first I will be using insulin. So the search take some time. So we have got the results for with a keyword search. For specific results, try to use the advanced search option. So this advanced search option provides us option to filter based on structure attributes, chemical attributes, sequence similarity, sequence motif, structure similarity, structure motif and chemical similarity. So let me open the first structure. So initial information we have classification of the protein, organisms of the protein and type of mutations. Further, when you observe here, there is experimental data snapshot which tells about the method the structure has been determined. So this method has been determined using solution NMR, that is NMR technique. Further, we also have macromolecular content, that is structural weight, atom count, other literature information. Followed by that, we have chain information. If there are any ligands also, the ligand information will be provided in the entity ID table. So let's see how a PDB format looks. So let's get those. So as we just mentioned, we have header section. So header section followed by the name of the protein, title, other molecular ID information. We have other remark based connect sections as well. Then here we see the atom section. So the atom section will give serial number of the atom, what is the atom, amino acid code, chain of the protein, residue number, x, y, z coordinates, occupancy and B factor. Again one more we have atom column has extra. So this is how for each atom in a protein a atom row is being assigned. Further, if we have any heteroatoms, so HCT is the code for heteroatoms and they are also named in similar fashion of the atoms of the protein. So we have terminal to differentiate between the chains and end to differentiate between the different proteins.
Excuse me, what does last column
It's B factor, but
tells?
if you see here, it is again an atoms only, but it's a simplified representations. Here if you take its C alpha carbonyl C, but here both are represented as C. This is simplified representation of atom, and this is protein structure based representation of the atom. So the same atom column, atom name column is repeated twice. Do you mean this column, right? Or this one?
Yes. Thank you. Thank you. I got. Thank you.
Let me move forward. So I have some questions here. So what does the PDB primarily store information about? So yeah. PDB stores information about three-dimensional structure of proteins, nucleic acids and other biomolecules. So now it's about section. So which section of PDB file contains a brief description of the structure, whether it's connect section, atom section, header section, or none of the above. Yeah, so it is header section, which contains the brief information, the title of the protein, experimental technique, organism, and mutation, any other details that required. So what information does the atom section of a PDB file typically contain? Yeah, so it contains atomic coordinates of each atom. So what does the connect section of a PDB file describe? So this connect section of a PDB file contains information about the bonds between the atoms in the structure, as well as other types of connectivity such as hydrogen bonds and disulfide bonds. Okay, now that we know, we have experimental techniques or computational techniques to generate the structures of three-dimensional structures of protein, or possibly quaternary structure as well, that is functional form of a protein, and we have a database to store it. But since the format and for 3D visualization there should be uniform, so we have some visualization tools. So here I have mentioned some of most popular ones. They are n number to use. Just give me a minute. Yeah. So the first is PyMol, so which is widely used. So it allows users to visualize and analyze protein structures in 3D and can generate high quality images and videos. PyMol is highly customizable because it has an option to plug in. We can also have some extra plugins, and PyMol compatible plugins will work with range of plugins and scripts available to extend its functionality. Then we have Chimera, that is Chimera X, which is next generation to PyMol, is designed for interactive exploration of large molecular sequences. So it includes a wide range of features, including support for virtual reality headsets, Then we have VMD. So VMD is again a new type or other molecular visualization tool. So this VMD analysis tool is designed for large-scale biomolecular systems. It includes advanced features such as supporting for multiple graphic card, interactive molecular dynamic simulations, and scripting capabilities. Then we have UCSF camera, so which is also again a tool designed for interactive exploration of 3D molecular structures. So it includes a wide range of features including support for multiple graphic cards and MD simulation. So other we have CN3D and JMOL. So JMOL is widely a Java based visualization tool that is designed for visualization and analysis of 3D molecular structure. So this JMOL is compatible with the many of the PHP based, HTML based and Apache based web servers. So in summary, there are many protein visualization tools available to researchers, each with its own strength and weakness. These tools are essential for the analysis and interpretation of the protein structures and are used in wide range of fields including structural biology, biochemistry and biophysics. The choice of tool depends on the specific needs of the user. The features and capabilities of each tool should be carefully considered before making the choice. So here next I wanted to move with the classifications. Just give me a minute, let me rejoin again. So proteins are diverse class of biological macromolecules that perform a wide range of functions in living organisms. Despite their diversity, proteins can be broadly classified into several structural classes based on their overall fold and topology. Here are some of the most common structural classes of proteins. It is alpha helical, beta barrel and alpha beta proteins. So alpha helix proteins have predominantly alpha helical secondary structure where protein peptide backbone is tightly coiled into a helix. So examples, we have some keratins and myoglobin. Then we have beta sheet proteins. So these proteins have predominantly beta sheet secondary structure where the protein peptide backbone forms the extended strands that are connected by hydrogen bonds. Then we have alpha and beta proteins. So these proteins have both alpha helical and beta sheet secondary structures. The alpha helical and beta sheets are arranged in distinct regions with the protein forming a distinct domain structure. So examples of alpha beta protein include thiophosphate phosphate isomerase or alcohol dehydrogenase. So in summary, proteins can be classified into several structural classes based on their overall fold and topology. Each class has its own distinct characteristics and function, and understanding these structural classes is essential for understanding the structure and function of proteins in living organisms. So we have two widely used classifications, COP and CATH, so which are widely used protein structure classification systems that provide a hierarchical organization of the protein structures based on their evolution and structural relationships. Yes, COP is a database, that is structural classification of proteins, that classifies proteins based on their structural and evolutionary relationships. The classification is hierarchical and is organized into 4 levels, that is class, fold, superfamily and family. So proteins are initially classified into one of the 7 broad classes based on their overall shape and architecture. Within each class, proteins are further classified into folds based on their secondary structure, content and arrangement. Further, folds are grouped into superfamilies based on their structural and functional similarity. Finally, each superfamily is further divided into families based on their sequence and functional similarities. For example, so we have protein lactate dehydrogenase, that PDB ID is 1LDN. So it is classified as shown. So its class is alpha and beta proteins. Fold is Roseman like alpha beta or alpha sandwich fold. Superfamily is lactate or malate dehydrogenase and family is lactase dehydrogenase like. So this is how SCOP classification looks like. Secondly, we have CATH. So CATH stands for class, architecture, topology and homologous superfamily. Is another hierarchical protein structure classification system that uses a combination of structure based and sequence based information to classify the proteins. Similarly, cath also have class, architecture, topology, and homologous superfamily. So proteins are initially classified into one of the four broad classes based on their overall shape and architecture. Within each class, proteins are further classified into architectures based on their overall shape and topology. Then architectures are further classified into topologies based on their secondary structure, content, and arrangement. Finally, each topology is grouped into homologous superfamilies based on their sequence and structural similarities. Let's consider the same example and how it is displayed, that is protein lactate dehydrogenase. So class is alpha beta protein, architecture is Rossmann fold, topology is NAD binding Rossmann fold domains, homologous superfamilies lactate malate dehydrogenase like. So in summary, both SCOP and cath provide hierarchical organization of protein structures based on their evolutionary and structural relationships. SCOP uses a combination of sequence and structural similarity to classify proteins, while cath uses a combination of structure based and sequence based information. Both systems are widely used in field of protein structure analysis and provides uh, valuable information. So let's have some questions. So after these set of questions, we'll move on about to the technique where how protein structure is computationally predicted. So SCOP and cath are two widely used protein classification. So how many hierarchical levels that SCOP utilized to classify proteins based on their structure and evolutionary relationships? Yeah. So again we have four: class, fold, superfamily, and family. So which type of the fold does the protein lactate dehydrogenase belong to according to SCOP? So what type of fold the protein lactate dehydrogenase belong to according to SCOP? So it is Rossmann like alpha beta or alpha beta sandwich fold. So before moving on to this, let's go. So now recently Nobel Prize has been awarded to AlphaFold. So let's move on to this server. So AlphaFold is a deep learning based protein structure prediction method. So this method uses multiple sequence alignment of the given input sequence with its homologous sequences and also use pairwise distance potentials from the given sequence. So this is how server interface looks. In a day we have a limit of 20 free jobs, and here one can predict the structure of protein, DNA, RNA, ligand and ion, and number of copies. So this copies comes in the situation of homomers, that is dimer, trimer, homotrimer, homodimer and others. So consider this given sequence. So I'll continue and preview the job. So it will take some time to predict. So the limitation of AlphaFold is sequence length of 4000. So it takes some time to predict.
So the limitation you said ki sequence of 4000. So 4000 could be matlab if we are looking for proteins, so 4000 amino acids, or if we are looking for DNA or RNA, so 4000 nucleotides.
Yeah, so that's what I'll explain then. Yeah, thank you for that question. For example, if I wanted to add a new entity again,
And
yes, please. Yes, please.
please go ahead.
Yeah, so here now again I wanted to check for any DNA, RNA, any. So overall number, you see the number annotation here. So each residue or each alphabet is counted as a token here. So this is the input. So when you give to so it's like 10 tokens still here. Sorry, it's going 20 tokens still here, 30 tokens. So it will take up to 4000 tokens to give. So if you have 1000 here, so in next input sequence you are only allowed to give 3000 tokens, that is 3000 residues. So overall structure prediction step, irrespective of number of entities, they only have 4000 token limit. Yeah, please, someone is having a question. Yeah.
And what does this copy means? Like
See,
copy.
if I click on copy, the number will increase. So if you want only single copy of this protein, so one chain will be built. If I increase copy, the homodimer of this will be built in the final structure. If we do this, homotrimer will be built. Actually, we will do that also in next demo sessions and see. Okay, as it is taking very long time, I will reduce some of the sequence length here and I'll delete it. So I'm increasing copy number to 2 because I have 20 free jobs to submit in a day.
So this 20 free jobs is different from what you just now told 4000 length.
Yeah, see, by default you can only predict structure of a protein, any protein, complex or single protein, single chain, multi chain, anything which has only 4000 atom connect. So it is limited to only to give atom information for only 4000 entries. Sorry, residue entries only 4000, not atom. Sorry, but this in
So
this
that means
each
4000
each
for
time.
one job. So similarly, 20 jobs can have 4000 into 20
Yeah,
residues.
yeah, you can
Matlab that I can.
yeah you can use 4000 in each 20. Yeah, one of the job is done. Result, let's view. So here we have PLDT, that is local difference distance score. So which is score used by AlphaFold to give clarity. Very high PLDT means a good structure. So it is a region based score. So blue color indicates this region is with high confidence prediction. Low PLDT means these regions are with less confidence. So this is how a two chain protein has been built, because we have given number of copies as 2. So further, let me try with 3 chains. Yeah, even in the recent concluded CASP 16, AlphaFold 3, their server version performed way better. Even our lab, Gromia lab, has been part of that. But still, we do see some disordered region prediction from AlphaFold 2. So if AlphaFold doesn't have a confidence in annotation for secondary structure, it gives a long loop like structures. So this AlphaFold copies method is feasible or applicable when you don't have chain information or the functional conformation information, that is whether it functions as a trimer, dimer, tetramer, or 18 chain protein, 12 chain protein, or like that. Okay, I will proceed with other methods. So we also have ESM fold. The reason why ESM fold is not that famous, because the limitation for ESM fold is single chain, so it can only predict one chain at a time. The residue limitation is only 400. Okay, let me copy paste some sequence from AlphaFold. But it is fast. ESM fold is little faster. I hope, yeah, we have tetramer result from AlphaFold. So this itself will identify the interface between these three chains and give. So this is the structure of the tetramer. Similarly, all the structures are classified, all the, all the residues in the structures are classified based on PLDTT score. Okay. So here the result is done. So along with this, we are also talking about PyMol, so which is widely used protein structure visualization tool. It will take some time. So this is the PyMol interface. So we can also visualize at atom level, residue level, chain level, molecule level, and only C alpha atoms. So when we observe on this left side, so we have options. So A stands for action. So zoom, center, origin, find, preset hydrogens and other activities. Then next after A we have yes, show. That means I want to show this structure as, okay, let me show sticks. Sticks will be displayed. Or if I want to show as licorice, some other different sticks. So before I showed hide cartoon as well. Show us licorice like this. Hide is to, so now that we have both the structures, I wanted to hide licorice and I wanted to show this structure as cartoon. Followed by that we have label. So label is to label residues. So residue numbers are added. L is for label. Or if I want to add atom names, B factor. So all the PDB information, PDB file information could be visualized here. So as of now, let me. Finally, we have color option. So I can color here. I can color them by chain, by element, by secondary structures, and by repetitions. So this is how a PyMol visualization tool is used. Further, we have plugin. By developing a suitable plugin, the plugin could be uploaded and it can be used. So PyMol plugin option is an extended version, like extended applicability version of the PyMol software. So similar structure. So these are JMol or RASMol based visualization. So on web interface itself, we can visualize the structure along with the sequence.
So excuse me, for the publication purpose, we have to download this, or how will it be? So suppose if I have a novel protein or something and I want to publish this, so I will go to any of the servers, for example, PyMol or something. So how will I will download, like, this is a 3D structure. So but for publication it will be 2D only. So just please throw some light on that.
Yeah. So here, when you go to the file option, this is in PyMol. So export structure, export image. So we have option. However, export a movie. Sorry, if you have any MD simulation, you can explore different conformations as a movie. Or if you need a structure for future use and you are not using it now, you can export the molecule. For this publication purpose, you have this PNG and all. And further, now you see the black background is black, but most of the publications we prefer a white background image. No. So here what we do is go to display, background white, then go to file. So it's up to you what background or what color combinations will enhance your image, like that. So if you, particularly if I want to talk about alpha helices, I wanted to highlight the color of alpha helices and have light colors for the remaining sheets. So this will highlight more. The reviewers or the readers paper will be easy. So even you can use secondary structure based coloring, sorry, yeah. So we have different coloring based, chain based coloring, secondary structure based coloring. Okay, I'll do it now. It's already there. Now the colors have been changed. So am I clear, or you want to anything more?
Yeah, yeah, no, thank you. So in this, like, it has 3D structure and I can rotate it any of the way. So whichever way I find it, matlab, everything is clear, that I will stop and I will
Yeah,
export as PNG.
yeah, PNG. So because for paper display purpose, many of the people use 2D representation only, but sometimes readers may request, for example, if your paper or a protein finds way good, or it might be you might have analyzed something in 3D perspective which I may not understand in 2D perspective. I may get back to you or write a mail back to you requesting, can you kindly provide your structure. And if the paper is published or the works are done, PDB has a deposit option. If you are feeling you have determined a very good structure than existing structure of the protein, you can deposit here. So you have different information. So based on this information, you can if your protein structure is novel, you can deposit here. So and just you can just provide a PDB ID. So reviewer or the readers just they'll type NL, sorry, LN, like this. So here the literature information, that is where this PDB structure is determined how and all is provided. Okay,
Okay, thanks.
thank you. Let me move forward. So now that we have seen, there are some other 2D representations that are used in representation of proteins, that is contact maps. So contact maps are graphical representation of pairwise interactions between the amino acids and protein structures. They are commonly used in protein structure prediction and analysis. So construction of a contact map barely involves two main steps. One is determination of pairwise distances between the amino acids, then is visualization of the contact map. So in this contact map construction, the first step is to calculate the pairwise distances between all pairs of amino acids in the protein structure. This can be done by using various methods such as Euclidean distance, Manhattan, or Pearson collision coefficient. So the distances are then converted into a binary matrix, just like a dot matrix what you were studying previously, where a value of 1 indicates distance between amino acid is below a certain threshold, and the value of 0 indicates that the distance is above a threshold. The second step is to visualize the binary matrix as a contact map, and this can be done by plotting the binary matrix as a heat map, where x-axis, y-axis represents the sequence position of the amino acid. So in addition to heat map, there are other visualization tools such as circular plots, 3D plots, and network diagrams. There are some softwares available for constructing contact maps such as Contact Map Visualizer, Contact Map Explorer, and Contact Map Analyzer. These tools provide various options for customizing the visualization of the contact map, such as adjusting the threshold for binary values, selecting the color scheme, and adding labels and annotations. So now that we can visualize the structure, but just by visualizing, can we tell number of hydrogen bonds, or how many hydrogen bonds, or which residues are forming a salt bridge? That is not possible. So we have some tools that provides inter-residue contact information. So before that, inter-residue contacts in protein refers to spatial relationship between amino acid residues in a protein structure. These contacts play a crucial role in determining folding and stability of the protein, as well as their interactions with other molecules. The different types of interresidue interactions include hydrogen bonds. These are formed between hydrogen atom of one amino acid and the electronegative atom, such as oxygen and nitrogen, of another amino acid. Hydrogen bonds are important for stabilizing secondary structures such as alpha helices and beta sheets. And there are So we have salt bridges also, which are electrostatic interactions between the oppositely charged amino acids. Salt bridges can play a critical role in protein-protein interactions and in stabilizing the tertiary structure of proteins. Then we have van der Waals, which are weak short-range interactions between the non-polar amino acids. So these van der Waals interactions helps to stabilize the packing of amino acids in protein core. Then finally we have disulfide bonds. So these are covalent bonds between two cysteine residues which can occur with a protein or between two different protein molecules. So disulfide bonds can play an important role in stabilizing the tertiary structure and quaternary structure of proteins. So this identification of interresidue contacts in proteins can be done using experimental techniques such as X-ray NMR. However, these methods may be time consuming and difficult, particularly for large or flexible proteins. Therefore, computational methods have been developed to predict the interresidue contacts from protein sequence or structures based on statistical or machine learning models. Further that before I'll show you that one of the tool. So we have short and medium and long range contacts in protein, so which refer to the different types of spatial relationship between amino acid residues in the protein structure. So short range contacts are typically defined as the contact between amino acid residues that are close in sequence, usually between few amino acids of each other. So these short range contacts are critical for forming and stabilizing secondary structures such as alpha helices, beta sheets. Then we have medium range contacts. These are the contacts between amino acid residues that are farther apart in sequence and usually separated by 4 to 10 amino acids. So these medium range are also important for stabilizing tertiary structure of the protein and typically form to the combination of hydrogen bonds, van der Waals interaction and electrostatic interaction. Then we have long range contacts. So these are the contacts between amino acid residues that are distant in sequence, usually separated by more than 10 amino acids. Long range contacts are critical for maintaining the overall structure function of a protein. They are typically formed with a combination of hydrogen bonds, van der Waals interactions, electrostatic, including salt bridges and disulfide. So yeah, let me show you one other method, that is PDB param. So PDB param method which gives you structural parameters of protein. So this works for all the protein that has a PDB ID. So let's look into its features first before accessing the tool. So here we have features divided into interresidue interactions, propensity, physicochemical properties and identification of DNA binding. So let's move on to interresidue interactions. What are the different interresidue interactions they provide? We have short range interactions, medium range interactions and long range interactions. Followed by that we have contact order, long range order, contact degree, 8 Armstrong contact number, 14 Armstrong contact number, total contact distance and multiple contact intakes. So provided this information, let's try to use the tool. So here they have given example PDB ID. So the example is 2TRX and chain name is A, and I wanted to select interresidue interactions. Further, it is asking what type of secondary interresidue interaction information we need. Okay, as of now, okay, let me select all and submit. Since we have selected all, it will take a minute or more just for the results to show, because it should calculate all these. So I'm repeating the steps again. So in the first compute, we have entered the PDB ID and chain ID. Then we have selected among the broad classes, that is propensity or DNA binding site, interresidue interactions or physiochemical properties. I have selected uh, inter, uh, interresidue interactions. Okay, I'll come back to the results when they have loaded. I'll proceed with these slides. Then we have solvent accessibility, surface area, so shortly known as ASA. So this refers to the portion of the protein surface that is accessible to solvents. sorry. ASA is important for understanding protein-protein interactions, ligand binding, and protein stability. So the calculation of ASA is typically based on the solvent excluded surface, that is CES model, which assumes that protein surface is formed by the boundary between the protein and surrounding solvent molecules. To calculate CES, that is solvent excluded surface, a rolling probe algorithm is often used. The rolling probe algorithm involves placing a probe sphere of given radius at every point on the protein surface, and then rolling the sphere along the surface to identify the points where the sphere first touches the protein. So the points where the protein probe touches the protein surface are considered to be the surface atoms, and the remaining atoms are considered to be the interior atoms. The ASA is then calculated by the summing up surface areas of all the surface atoms. The surface area of each atom is determined by the area of the sphere that is not covered by the protein. But still, that for a human visualization or a human, it's impossible. So we have methods like NAccess, DSSP, NetSurf, RVPNet, and Scratch to determine. So of which DSSP and NAccess are the widely used softwares for calculating solvent accessibility of protein. So where they use rolling probe algorithm to calculate the solvent accessible CES area. Among them, DSSP also, along with the solvent accessible surface area, it also provides annotation for the secondary structure information. I hope results are done. Okay, yeah, let me reduce some. So followed by that, we have one measure which is an indirect measure for the local compactness of protein structure, and it is defined as the average sequence separation, that is contact order, which defines the average sequence separation between the amino acid residues that are contact with each other. So it is an important predictor for folding rates of proteins, and proteins with lower contact order tend to fold faster than with the higher contact order. So because this contact order, as indirect measures are developed for the machine learning algorithms, where these values are model trains to use for uh, make new predictions. So the formula for contact order is defined as separation between the residues I and j divided by total number of contacts in a protein and total number of residues. Further, we have similar measure, so which tells about long range order. Sorry. So long range order is also similar, which tells about the interactions that are close in space and far in sequence. So I have some problems on both of them. Let's try to solve. So given the solvent accessibility of residues alanine 7, glutamic acid 8, valine 9.
Excuse me, your screen is not visible. Your screen is not visible.
It is visible, right? I can see in my other system. I can see my screen. You want me to reshare again? I'll do reshare.
Yes, please.
Now,
No, again, it is not.
because in my other system where I'm logged in, I can see the screen there.
No, till now it was visible, but from this slide only, the previous slide, it is not.
Now, if not, can you kindly rejoin again from your system?
Now it came assignment quiz, no?
Yeah, yeah, okay. Thank you. That works. So given the solvent accessibility of residues, so based on the solvent accessibilities, what types of which are the residues that are located at the surface? So, residues which are located on the surface have higher solvent accessibility. So in these residues we have glutamic acid 8 and alanine 11, so which have values of 56 and 78 Armstrong square. So these residues are considered as the surface residues. Next I have the accessibility of a residue arginine is 74 in folded and extended state. So what is the percentage of accessibility? So given individual accessibilities in folded and extended state, so what is the percentage of accessibility? So the percentage of accessibility is calculated as folded state accessibility divided by extended state accessibility. Okay, it is 47.2%. It is folded state, that is 78 divided by 165 into 100 to convert it to, this is the accessibility formula. So next I have a question. A protein has 20 alanine residues and 5 are on the surface. So what will be the buriedness of alanine? So the formula for buriedness of alanine is given as number of residues of I in the interior of a protein and total number of residues. For example, since I mentioned alanine, so calculate it as number of alanines in the interior of the protein divided by the total number of alanines. This gives the buriedness of a protein. So number of, first numerator is number of alanines in interior of the protein, but here I have mentioned out of 20 alanine residues 5 of them are surface. Then how many number of residues are available in the interior? Divide by total number of alanines in the protein, that is 20 by default because I have given it. Yeah, so here so since we have out of 20 alanine residues 5 of them are surface, so we have 15. So 15 divided by 20 gives the buriedness of the alanine. So I have next question. A protein has similar 20 alanines on residues and 5 of them on surface. What is the transfer free energy? So to get this transfer free energy, partition coefficient has to be computed. So which is ratio of occurrences of residue to be in the interior to the of that protein, and further this is RT ln buriedness. So RT ln buriedness will give partition coefficient of a protein. Can you try calculating? I'll wait for a minute here. Since I have given already the value for buriedness, try to calculate it for here we have R value of 1.98 kilocal per mole per Kelvin. Temperature is 300 into further ln of 0. Log of log. So there is some calculation mistake. I'll correct it again. So but it is, the formula is minus R, so minus 1.98 into 300 into log of 0.75, that is 15 by 20. So next I have fourth question. This is regarding the contact order. A protein has 50 residues and 200 contacts. Residue alanine 45 has a contacts with residue 46, 47, 44, 71 and 60. So what is the contact order of the residue 45? So this is the formula for contact order. So where Sij is the separation between the residues. Okay, so let's start calculating it. So we have denominator L, that is total number of residues in the protein. We have 50 residues. These 50 residues are forming n contacts, that is 200 contacts. So the denominator is 50 divided 50 into n. The numerator is separation. So the alanine between the separation between 45 and 46 is 1, plus separation here is with glycine 47 is 2, 1 plus 2. Again with alanine 45 and alanine 44 is 1. So 1 plus 2 plus 1 plus 16 plus plus 26 plus 15. So this is the numerator. So when you divide this separation with 50 into 200, the value we get is 0.045. One separation is 1, 2, 1. So summation of Sij will come to 45, and the numerator is 200 into 50. So overall the contact order of a residue alanine 45 with these given neighboring residues is 0.0045. Then a similar one, so on the similar protein we will try to calculate long range order. So this is the formula for long range order. I'll wait for a minute, you guys try. So here we have a binary numerator. So the numerator binary value will be 1 if the distance between two residues is more than 12, otherwise it is 0. Rest remaining are the same. So here with more than 12 sequence separation, we have only residues valine and isoleucine. So we have now 2 divided by 50. So it is 0.04. Further, we calculate the surrounding hydrophobicity. So here I am asking surrounding hydrophobicity for the residue threonine 25, which is surrounded by 2 alanines, valine, 1 valine, 2 threonines, 1 glutamic acid and 1 serine. So the hydrophobicity values of individual amino acids are given. So this is the formula for hydrophobicity. Try to calculate. Since today I don't have access to my right pad, the detailed solved solutions I'll be uploading along with this PDF of the PPT. So for alanine, the value is so if we take alanines 2, so 2 into surrounding hydrophobicity of alanine plus valines 1 into surrounding hydrophobicity value of valine plus 2 into surrounding hydrophobicity of threonine plus 2 into surrounding hydrophobicity of glutamic acid plus 1 into surrounding hydrophobicity of serine will give you a value of -3, which is the surrounding hydrophobicity of the residue threonine 27. So here I have one more question. So which method provides the information on both secondary structure and solvent accessibility? As I mentioned, DSSP, which is Dictionary of Secondary Structure of Proteins. So this will provide information on both secondary structure and solvent accessibility. Next, pi mole. So this is pi mole can be used for, so whether the pi mole can be used for protein structure visualization or for computing bond length, bond angles or torsional angles, or to prepare high quality figures, or all of those. So pi mole is the protein structure visualization tool, and also used by using the plugins. It can compute bond length, bond angles and torsional angles, and it can also prepare high quality figures. All this we have discussed in the pi mole section. So before that, this page has some issues with the result, but to access a PDB param, a detailed tutorial has been prepared. So this tutorial will help you how to provide input to the PDB param, and what are the options to select, and then how a sample output looks like. So the sample output is also similarly planned as a tabular output that follows the PDB format. So we have residue name, residue number, atom name, chain name, residue name. Again, this is for identification of protein binding site. So in terms of contacts, we see here a residue name, glutamic acid at residue position 2 in chain A has number of contacting residues of 5. So these residues are glutamine 3, arginine 4, threonine 43, isoleucine 44 and asparagine 45. Like this, individual interresidue interaction information will be provided in the PDB param page, and the formulas on which PDB param has been constructed are provided under features, interresidue, so the detailed explanation along with the reference of contact order, detailed explanation of the formula of long range order along with the reference, contact degree, 8 Armstrong contact number, 14 Armstrong contact number, total contact distance, multiple contact index information. So all these formulas along with their references has been provided here. So in this class we have discussed about AlphaFold, but most of the times if we have a sequence, the information we know is uniprot. So okay, let me take any uniprot ID. Let me consider the and one more disadvantage here is, for a given sequence which is widely studied, n number of structures are available. So how to access all those structures? So under this structure tab, we have all the information of the structures available for a given uniprot ID. So here we see, for this given uniprot ID we have different structures covering a different range. The first structure, which is 1AP, which is of X-ray, has a resolution of 1.05 Armstrongs and has a chain A and B. It has information from residues 287 to 344. Further, if we scroll down, we have some NMR structures. Lastly, if you scroll to the last we have AlphaFold predictor structure as well. So AlphaFold has already uniprot has hyperlinks to the many of the AlphaFold predictor structures, and this structure can be easily downloaded or accessible from this download link given. It will redirect in some time. Yeah, so we can download a PDB file, download, then open it through PyMon, and next are the same steps. Yeah, so I have that's all for today. Today I couldn't access my write pad, so I couldn't solve step by step of these problems, but I'll provide solutions in slides. So I have from previous year. I'll also provide a link to my previous year video where I have solved similar problems in last year lectures. So if any doubt, I'll be happy to address. If not, you are free to leave. Thank you for attending despite preponement of the class. So here further I have added some more questions along with the detailed explanation of the X-ray methods. Yeah, because this is a we have continuation lecture on next week as well. So the more detailed solution of these lectures will be discussed in the next upcoming class. So here I have a question on temperature, B factor, occupancy, buriedness, and experimental techniques for structure prediction. About packing density, transfer of energy, contact order, long range order, other things. So do try to solve them before the next class so that it could be easy to start the explanation again. Yeah, and also regarding, sorry, I forgot to mention, regarding the download, PDB allows you to download in FASTA format, PDBX or MMC format, which is other format visualization, and you also have PDB format, validation format, and biological assembly format. And PDB also has different tabs for structure, annotation, experiment, sequence, genome, and versions. So we have different information under different tabs. As I mentioned, if we have any of the new data, it could be deposited using the deposit option of PDB. So here it tells what is the deposition date, so deposited by authors, and also revision version. This PDBs accept to have revisions from the other authors about the structure, and the revision history will also be posted. So that is followed by initial revision. For this insulin, we have version format complaints 2 times. Then we also have derived calculations and other polymer sequence. Finally, recently we have data collection changes, data reference changes, and structure summary changes added to this. That's all from my side. Thank you for attending.